Drawing on some previous genomics work from members of the lab, there does seem to be population structure in Xylella in California, meaning that there's different populations that seem to group by where they're from. So you can see in the diagram on the left, there's different groupings from strains in Napa, in Sonoma, and then in Southern California. And there are specific SNPs, or single nucleotide polymorphisms or mutations, in the genome that do seem to be associated with temperature. So some mutations might be associated with colder climates and warmer climates. So there is already evidence that Xylella there are parts of the genome that are related to temperature. Drawing on this, you'll notice that in the original data set, there were no strains from Northern California. And what a farm advisor, Glenn McGorty, had noticed was that Pierce's disease was increasing in Northern California in Hopland, which is in Mendocino County, as you can see on the map on the right. And so with this increasing incidence of Pierce's disease, we started to wonder about local adaptation in Northern California. And so in the fall of 2020, we sampled a population of Xylella strains from Hopland with the hypothesis that these strains might be adapting to colder climates because this area in Hopland was on the edge of where Pierce's disease had been because of the colder temperatures there. And so like I hinted at in the last slide, the reason that there wasn't much Pierce's disease in Northern California, at least so we thought, was because of this overwinter recovery related to winter temperatures. For a long time, it was established that over 80% of vines with Xylella will recover when they're inoculated after May. And this overwinter recovery does appear to be temperature linked, but it's unclear whether it's the bacterium responding to colder temperatures in the winter, or if there's an indirect response, meaning that something's happening in the plant and that plant response causes the disease to go away over the winter. And so if there is a plant response, there may also be varietal differences in this overwinter recovery. But with climate change and this increasing incidence of Pierce's disease in Northern California, we thought this might be related to climate change. And so here on the left, you can see the minimum annual temperature in California over the last 65 years. And the two circles are, the top one is Hopland, California, where we sampled this population, and then also in Napa. And they're also graphed on the right, so you can see the minimum annual temperature and a moving average. As you can see now, it's clear that those have been increasing from around 1990. It's also clear that Xylella in Hopland and in Napa have very different environments to adapt to. So even though they're pretty geographically close together, in Hopland, the annual minimum temperature is closer to 4 degrees, and in Napa, it's 3 degrees warmer. Given that there is clear evidence of climate change and how this affects Xylella, we were wondering how this would affect Pierce's disease in Hopland and what happens between different varieties. One of my colleagues in the Almeida lab, Alexandra Kahn, had started to ask some of these questions with some of her previous work. In her work, she did a multi-year field experiment where 3 different varieties, Chardonnay, Merlot, and Cabernet, were inoculated with Xylella. And she found that there were variety-specific differences in recovery rate. And what you see here is the proportion of vines that were positive. And the Chardonnay line, which is in red, seems to have lower overwinter recovery, because as you can see in 2009, it has a higher incidence of Pierce's disease. And then also, there was a lot of surprising data about what symptoms were observed in the first season versus the later 2 seasons. What you saw in the first season was that there was a lot of shriveled clusters, which is the photo on the right. And that was the first symptom that was observed, which was a little unexpected, versus in the later season, there was a lot of stunting earlier on, and then more foliar symptoms later in the season. And what was learned from this was that the symptoms may not be exactly what we expect because it's hard to observe vines and infections under field conditions. So keeping all these questions in mind, we had a unique opportunity to study some of this with this experimental vineyard in Hopland. This is a site that was set up at the Hopland Research and Extension Center to look at 13 different Mediterranean varieties in California climate. So each of the varieties, there are 30 to 40 vines planted, and this was all planted around 7 years ago. So at this point, they are mature vines. And in the fall of 2019, all of the vines were tested for Pierce's disease, and they were all negative. So that kind of set the stage to do some inoculation experiments. And with this project, we had 3 different research objectives. The first, the genomics arm of this, was to look at local adaptation of Hopland strains to climate in Northern California. Next, to understand the progression and movement of Pierce's disease in different varieties under field conditions, because this plot has a great opportunity to do comparative studies, we can compare how Xylella moves and progresses between varieties, and then finally to look at overwinter recovery rates of Pierce's disease in different varieties, but also to use two distinct strains, meaning we'll have one strain that's from Hopland and hypothetically adapted to the colder climate, and one strain that's from a warmer climate in Southern California. So here are some preliminary results from the genomics part. We have a phylogeny that shows clusters of populations of Xylella in California. And what was a little bit surprising is that the strains that we sequenced from Hopland kind of split. And one group formed this Hopland-only clade, which is in purple at the top of the page. And then there was also a group that was Hopland strains and strains from Sonoma that were clustered together. So it seems that there are two distinct genotypes coexisting in Hopland. And so we can look at those, start to delve into those more. Next. So for the field experiment, I said we wanted to compare two different strains from two distinct climates. And so for the field study, we selected one strain from the Hopland-only clade, with the thinking being that this would be cold adapted, and then one strain from Temecula, which is color-coded in yellow here. So here you can see our experimental design. We had control plants, which were just inoculated with buffer, and then cold strains. So that was the strain that was from Hopland, and then the hot treatments. And so these were randomized within all of the 13 different cultivars. Next. Here is our sampling scheme. In May of last year, we inoculated 481 vines, which was a big field day, and we inoculated all of them on the western shoot so we could keep track of movement across the vine. In June and July, we selected 3 varieties to sample at 4 locations across the vine. And so we selected 3 just because of the size of this plot, so that we could subsample those to really understand movement of the bacterium across the vine. Then in August and September, we collected observations of symptoms on all 13 varieties. And then in September, we sampled the western shoot on 10 varieties. And then for the 3 varieties for which we had been doing this all summer, we sampled across the vine to complete that data set. So that's what we were able to do in the first year. So just to give you a little sense of what this looks like, I had a lot of help from my amazing lab team, who's shown here, helping me inoculate 480 vines. Then in September, I had a lot of help also from some of my undergraduate students and everyone in my lab to help collect these samples, label all of these samples, and to start processing all of these. Some preliminary results. We observed a range of symptoms in the first season, including shriveled clusters early on, and then stunting and a lot of leaf scorch symptoms. And as you can see from this diagram, the symptoms that we observed, the percentage of symptomatic vines, did seem to vary across cultivars. So cultivars like Tinta Francisca, which is the yellow line, had a higher incidence of symptoms than others like Terrell Dago. So this plot kind of showed dots for where the symptoms were in the field, but the point is that there were a lot of different symptoms that we observed across different varieties. Then just to show you what the symptoms look like, in this variety, Graciano, we see this scorching on the leaf edge and the bright red coloration, and then the western shoot, which is the photo on the left, a lot of the leaves do seem to be dried out, and you can see scorching symptoms there too. And then in another cultivar, Tinta Amarella, there's the western shoot, which is the inoculated one. It seems to be totally dried out. And then I included a close-up of the shriveled clusters, which we observed. We also saw this symptom in one of the varieties that seemed to be a little different and new, is that the inoculated shoot didn't have any leaves. It was completely defoliated or barren. And we saw this on multiple vines in this cultivar. And then also you see the shriveled clusters are very obvious in the photo on the right, but we didn't see this in other varieties really. So it seemed to be unique to this one that the inoculated shoot was completely defoliated. As far as testing, which vines are positive for Xylella by qPCR, in July, you can see some of the results that we only sampled a few cultivars. So those are scattered around a bit. And in September, for all of those samples, we've been able to process over 300, so up through row 10, but we still have a lot more to go. And you can see, I think, 2 controls or 3 that were positive so far, and it's unclear. We'll follow up with that to figure out if that was an error in the inoculation or the sampling or in the molecular processing. And so from the qPCR results on the left, you can see the 13 different cultivars and the percentage positive in the cold, control, and hot groups in July and in September. And this is preliminary data as we're still testing, but I initially fit a binomial response generalized linear model. So whether the vine was positive or negative with treatment, so cold or hot, and variety as predictor variables. And what we've seen is that there hasn't been a significant difference between the strain, so whether it was the hot adapted or cold adapted strain in the infection response, but there is a significant difference between the different varieties in infection rate, both in July and in September. In conclusion, we are seeing that there are varietal differences in the symptom expression and in susceptibility by qPCR, and we'll continue to fill out that data set and also process some of the samples that were across the vine. So far, there don't seem to be any differences between the strains in infection rate in the first year, but we may see differences in overwinter recovery, and that we'll be able to tell starting next year. So there's a lot of work with this project that's still in progress. So the genomics analysis of genes involved in cold tolerance, as I said, looking at movement across the vine. And then there's plenty of new statistical models for symptom progression and colonization that we'll build from this project. So I would just like to thank everyone in my lab who helped with the field work, and my 2 excellent undergraduate students, Eamon and Josie, who helped with the lab work. Obviously, none of this would have been possible without a large team of people to help with the massive effort of collecting samples and processing samples.